Okay, so I think we'll get it started. Good afternoon, everybody. Welcome to the SWOG Immunotherapeutics Committee meeting. My name is Siwen Huliskoven from Huntsman Cancer Institute, University of Utah. Oops. And, I co-chair the Immunotherapeutic Committee with Dr. Katie Politi from Yale University. She's joining us online today. So we have a full agenda, and this is a virtual, a hybrid meeting. So I want to remind the attendees online, please just mute yourself, turn off your microphone. And also we have speakers who will present virtually. So you should be able to share your screen when it's the time. So if you cannot do that, please let us know as soon as possible. We have a full agenda. So Dr. Aaron Ring from the Yale University will give us an interesting talk on autoantibodies and the response to immunotherapy. And then we will have a session where we invite the IO leads from the cross NCTN cooperative groups to showcase their efforts in their groups. And in the end of that session, we'll have a discussion, to discuss challenges, success, and ways to collaborate. And then we will have our lead statistician, Dr. Michael LeBlanc, to speak on how to utilize SWOG data for immunotherapeutic studies. And in the end, we'll provide an update on IMATCH pilot. We will have an in-depth discussion with our study team, and several CRC from the high enrolling sites is going to speak, share their experience, and answer questions. So without further delay, I want to invite my co-chair, Dr. Politi, to introduce the first speaker.
Hi, thank you very much, Dr. Huliskoven. I just wanted to check if Dr. Ring is online before going ahead with the introductions, because I do not see him in the list of presenters. Maybe he hasn't had a chance to join quite yet.
I don't see him.
So I wonder if we should move on to the next item on the agenda.
Sounds good.
I will email in the meantime.
Sounds good, Katie. So let me just fast forward. So the SWOG leadership actually is among the first to realize immunotherapeutics is an independent identity from all the different disease groups and needs a separate committee to support the efforts. So since our IO committee has been established 3 years ago, Katie and I have been experimenting different ways to support our investigators. So, and we are also curious how this is executed in other collaborative groups. So today we have IO leaders from Alliance and NRG to share their experience. And we also have Dr. Ignacio Vistuba. So there's no independent clinical IO group within ECOG, but Dr. Vistuba actually leads a IO biomarker group at ECOG, and also he leads the CMAC effort. So he's going to talk about that today. So I'm going to invite Dr. Politi to talk about our SWOG IO committee initiatives.
Thank you. Thank you very much, Siwen. I actually see that Dr. Ring has joined us. How would you like to go ahead? Should we have Dr. Ring present first and then jump to this?
Yeah.
I think we're still, close enough at time. Hi, Dr. Ring. How are you? Thank you very much for joining us. Maybe we'll just go to his presentation, and then we can go back to the second item on the agenda.
Yeah, that's okay. Is Dr. Ring ready?
Sure. Yeah. Apologies to everyone for not putting the Zoom. I didn't put the Zoom link in my invite. I was frantically searching my email for it.
No problem, no problem. So I'm going to go ahead and introduce you just a second. So today, it really is my pleasure to introduce Dr. Aaron Ring, who's an associate professor of immunobiology at the Yale School of Medicine. And he's going to tell us about work that he's been doing on studying autoantibodies and response to therapy. By way of introduction, Dr. Ring is a Yale undergrad who did then an MD PhD at Stanford University. He works on protein engineering and systems immunology and has continued that work in his own lab, which he started in 2016 in the Yale Immunobiology department. And I think, I think an exciting piece of news, at least in terms of SWOG and collaborations with SWOG, sad for those of us at Yale, is that actually Dr. Ring is going to be moving to the Fred Hutchinson Cancer Research Center. But that also is a SWOG institution. So hopefully there are opportunities to collaborate and continue to work together. So really happy to have you here today and excited to hear about your work. Thank you.
Yeah. Thanks so much, Katie. It's really a pleasure to be here. Is my screen right, or shall I swap displays? Is that looking okay?
If you could swap, that would be great.
You got it. There it is. Okay.
Thank you.
Yeah. So I'll dive right into it. Look, as Katie said, I'm a protein engineer. That's what I've worked on most of my career. And these are my disclosures that really reflect that I make drugs and have been involved in translating them into the clinic. But related to my talk today, in founding this company, Cerenova Bio, it really reflects a shift in our research interests and priorities, which is that I've made a sad realization, midlife crisis, if you will, that all of my skill as a protein engineer is not what's rate limiting to developing new therapeutics that are going to be transformational patients. Rather, it's about knowing what to drug and when to drug it. It's all about targets, right? Making drugs is actually downhill skiing. And so the work that I'll present today reflects my lab's efforts to throw our hat in the ring and see if we can generate unique insights into what's important in cancer immunotherapy. And the idea that we had is that instead of engineering drugs to interrogate the immune system, why don't we do the opposite? Why don't we determine the impact of the natural drug products of the immune system on patient health and disease outcomes? Essentially, we want to think about patient autoantibodies, which are essentially natural biologic drugs, as major contributors to phenotypic variation between patients. And you can imagine that there are antibodies that make things worse, that are harmful. And this doesn't take a lot of imagination. These are typically what people think about when they think about autoantibodies, think about those that are etiologic in autoimmune disease. But there are many examples of autoantibodies that are actually beneficial. And cancer is a great example. It's been known for decades that patients that make antibodies against their tumors have better responses, better survival than those that don't. Like, for example, anti-HER2 in breast cancer, anti-MUC1 in different carcinomas. Lloyd Old himself kicked this off with his CLEX technology and found a number of tumor-associated antigens, many of which are being targeted today. So really the premise of our research is that autoantibodies can teach us about what to drug. We just have to know what drugs were present in a patient. We can sort of decode these clinical trials of nature. And so we thought, okay, well, autoantibodies can target a lot of proteins. There's tens of thousands of proteins in the proteome. Can we reduce our search? And our idea was, you know what, we don't have to look at the whole proteome. We can target the extracellular proteome. And that's because antibodies are themselves large secreted proteins. They exist outside the cell. And there they can exert biological impacts through a number of different mechanisms. They can directly block or activate signaling through receptors, or they can target cells or molecules for degradation or destruction, for example, through ADCC or fixing complement, et cetera. The problem is that these proteins, these extracellular proteins, are among the most difficult to sample. They have unique folding requirements like disulfide bond formation, signal peptide removal, glycosylation, et cetera, that make them difficult to sample in their natural three-dimensionally folded conformation. And that's critical because 90% of antibodies recognize antigens that are conformational rather than linear. So to approach this problem, we look to create a platform that will allow us to scan the entire human extracellular proteome for antibodies, but also preserve as much as possible this native folding. So our solution to this problem was to turn to our friend, the trusty baker's yeast. And it turns out that yeast are pretty good at making our proteins. And I knew that because I've been working on using yeast display to engineer, immunoreceptors and ligands for almost 15 years now. And so we made a big bet that we could capture most of the human extracellular proteome on yeast. So what we've done is we've painstakingly curated a library now of around 6,500 unique proteins displayed clonally on the surface of yeast. So each yeast has one barcode. And to do this approach, we call it REAP, Rapid Extracellular Antigen Profiling. We just mix together the yeast library with a little bit of patient sample, about 50, 100 microliters at most, magnetically isolate the antibody-coded clones, and then we just sequence the barcodes. So we turn an antigen-antibody binding event into an ultra-high throughput, multiplexed sequencing readout. And this enables us to have, quantitative, deep, and high throughput assessments of what autoantibodies are present in patients. At this point, we've now profiled over 6,000 samples from over 4,000 different patients across a wide spectrum of disease indications. And what you can see when you cluster the data is really interesting. We actually see that that autoantibody signatures define clusters that group patients by states of health and disease. So you can see that healthy individuals tend to cluster separately from those with acute COVID-19 or ME/CFS or metastatic cancer in this case, or RA, psoriasis, lupus, et cetera. So that was pretty interesting to us. But related to cancer, we had a specific question, which is, do autoantibodies impact response to immunotherapy? And the idea here was, well, in the same way that genes can influence response to drugs through pharmacogenomics, might there be a pharmacoceromic paradigm, if you will? Might some patients have autoantibodies that can enhance response to therapy? Might others have antibodies that inhibit response to therapy? And this could teach us a lot. Antibodies that enhance immunotherapy are telling us about drug targets, right? They could be immunomodulatory, they could be tumor opsonizing. On the other hand, some patients have antibodies that inhibit response to therapy. That's telling us about essential pathways that are needed for immunotherapy. This could also lead to development of biomarkers. So we teamed up with my colleague at Yale here, Harriet Kluger, and also to look at about 250 patients, looking pre and post treatment with immunotherapy, either anti-PD-1 monotherapy or the combination of CTLA-4. Some of these patients we actually followed over 2 years. And then we also supplement that with additional samples from another cohort from a commercial biorepository. So in total, we have about 400 patients in this cohort. And right off the bat, just globally, I can tell you that cancer patients have just a remarkable diversity of autoantibodies in their blood. In just 400 patients, we found almost 3,000 unique autoreactivities. And in fact, we haven't even completely determined the full set of autoantibodies that are present. If you do a discovery rate analysis, you can see with every incremental patient that we add, we're finding new unique reactivities. We've basically discovered all of the common reactivities, antibodies that are present in more than 1% of the population. But we are still continuing to discover rare autoantibodies. Again, like genetics, you see, some alleles are common, others are rare. Those rare ones can actually have a big impact. So we need to go bigger. And the other thing I can tell you, like I said, is that most autoantibodies actually end up being rare. Half of all antibodies we only saw in a single patient, right? And that most autoantibodies were present in less than 1%. So clearly, we need to go bigger with a bigger cohort. But we can still make a lot of conclusions. So first thing that's interesting globally is that cancer patients, even prior to treatment, have an elevated number of autoantibodies relative to healthy. And this was basically consistent across many different types of immunogenic cancer. All of these patients got anti-PD-1, so I assume all immunogenic. So that was interesting. So then the question becomes, okay, well, what about response to therapy? And this also really surprised me. Turns out that treatment with checkpoint inhibitors didn't really change the number of autoantibody reactivities we saw. On average, patients who got anti-PD-1 or anti-PD-L1 therapy had no net increase in the number of hits. Okay, as many patients lost hits as gained them, and the mode was 0. If you add CTLA-4 blockade on top of that, the average number of hits now goes to 1. You get 1 new hit on average if you add CTLA-4. The mode is still 0, and some patients lost, but you can see there are more patients who gained autoantibodies than lost them. So really, what we found is that autoantibodies are pretty stable over the course of therapy over a long duration, and that if you look at the survival of an autoantibody in patients, and this is aggregate for every autoantibody we detected in every patient, you can see that 50% of autoantibodies we saw prior to treatment are still present in some patients more than 600 days later. Okay, so really, we can think about autoantibodies, like I said, almost like genes. And that's how we've started analyzing the data. We calculate odds ratios. We say, okay, for every autoantibody we saw, what are the odds that you had a response divided by the odds of having a response if you didn't have that autoantibody? So just like the gene, we calculate the odds of a response based on having an antibody with some adjustments for, what type of therapy were you getting? Was it the combination? Was it the monotherapy? And what type of cancer? We're obviously more impressed for a response in a lung cancer than a melanoma. And so we need to adjust the odds depending on what we see. And then you can, create these types of familiar volcano plots you're used to seeing, where you can plot the, measured effect size of having an antibody versus its statistical significance. And antibodies in this upper right-hand quadrant, these are antibodies that are presumably treatment enhancing. They're associated with the response. They're telling us about potential drug targets. Over here, we see antibodies that are associated with progression. And maybe we should do the opposite and try to activate these pathways. So I'll just jump right into it. We've seen some really quite fascinating examples of antibodies that can enhance immunotherapy. And one blazing signal we saw was our old friend, anti-type 1 interferon. It turns out that about 3 to 6% of the cohort has these anti-interferon antibodies, and they're associated with very high odds of responding to therapy. Odds ratio is about 5 to 10 if you have these antibodies. And that was really surprising to us. We'd seen these before in the setting of COVID-19. Turns out, these are heroes in cancer, absolute villains in COVID. Your odds of dying of COVID-19 if you have anti-interferon are about 20 to 200 times higher than if you don't have those antibodies. So they're actually associated with about 20% of all COVID-19 deaths. But here, they're associated with much, much better responses to immunotherapy. And we've actually studied these patient antibodies. We looked to see what their function was. And you can see that in general, these antibodies potently neutralize interferon signaling in ex vivo assays. Interestingly, we found a couple patients who progressed, who had these autoantibodies, but their antibodies did not neutralize interferon signaling. So that really allowed us to focus in on the activity as interferon neutralization. And that didn't make sense to me at first, but if I'd been a better immunologist, I would have known about Andy Min's work, which we eventually found, which really was, the Rosetta Stone in understanding what these antibodies meant. Turns out, that I had thought interferon was amazing at shrinking tumors, and historically it is. It's really important, for promoting antigenicity, adjuvanticity of immunotherapy. But the cutting edge of the biology that Andy's group has shown, but also additional lines of evidence from John Taharo, and now objective data from Dan Manguso at Dana-Farber, is that chronic low-level interferon signaling promotes potent immunosuppression due to upregulation of ISGs, including PD-L1, and that genetic deletion of type 1 interferon signaling through like IFNAR deletion, et cetera, actually increases the sensitivity of animals to immunotherapy. But we tested this pharmacologically, and across multiple different tumor models, we found that addition of interferon pathway blockade, either through IFNAR and anti-IFNAR antibody or a anti-interferon antibody, that we could enhance checkpoint immunotherapy responses, for example, in B16F10 or in the CT26 model, consistent with what we saw in patients and what Andy and others have seen mechanistically. So this was a great proof of concept to us that these treatment enhancing autoantibodies exist, and that we can find them through this approach. But I want to tell you that it's not just interferon. This theme that patients can make autoantibodies to cytokines that drive pathological inflammation is actually fairly prevalent. And we see examples of patients who make antibodies to IL-6 and IL-17 and LIF family receptor cytokines, and they generally have higher odds of responding than if you don't have that. And, it turns out that just last year, independently, another group found that blocking interleukin-6 enhances responses to immunotherapy preclinically. And we found, again, like interferon, that these patient antibodies neutralize IL-6. So, again, just, a further validation that that this paradigm of neutralizing pathological cytokines can produce benefit in response to therapy. But it wasn't just cytokine autoantibodies. We saw other things that were pretty interesting were that patients who responded to therapy tended to make autoantibodies to tumor-expressed antigens. And so in blue are examples of actual, validated tumor-associated antigens like mesothelin, Claudin 18. I got pretty excited when I saw EGFR here. It turns out these are actually not autoantibodies. These are lung cancer patients who had gotten cetuximab like 6 years prior. So this is actual drug we're measuring. But in general, these antibodies are associated with better outcomes, consistent with what people had seen with HER2, et cetera. But all those red dots are sort of novel, prospective TAAs where we found patients who made antibodies from tumor-expressed antigens, and they did better. And in general, these tend to be opsonizing isotypes like IgG1 and IgG3, and they elicit ADCC. So we're working these up as next generation targets for bispecifics, CARs, ADCs, et cetera. But I want to end, just on a note. We've seen a lot of exciting stuff, but we're just scratching the tip of the iceberg. And we can finally, now having run, about 5,000 patient samples, do a power calculation because we know the frequency distribution of autoantibodies in the population. And I told you that the average autoantibody is rare. In fact, we now know that the average autoantibody is present in about 1 in 500 individuals. Knowing that, we actually know that with our current cohort size, we're only powered to detect associations for autoantibodies that are relatively common, that are present in more than 1.5% of the population. And so we need to go deeper. So this is really a plea to all of you. If you have these types of interesting samples where you have patients who got checkpoint immunotherapy and you have plasma or sera on those patients and their radiographic response, it would be amazing if we could add them to our cohort and allow us to, shift to the left and be able to detect even more, clinical associations than we've seen so far. And that may allow us to bring, some really interesting targets into focus. Sorry, I don't have time to present this slide. I should probably just go to my conclusions here. So just to wrap up, hopefully I've convinced you that studying autoantibodies can point us to important potential targets that, by virtue of the causal effects these antibodies are exerting, that there's incredible diversity of autoantibodies in cancer patients, and that these autoantibodies do partition patients by responders and non-responders. We've been able to validate some of these insights preclinically, and we're really hopeful that we can, increase the power of our studies by augmenting the cohorts that we've collected so far. So I'd just like to briefly wrap up by thanking the folks who have been responsible for these efforts. So my own lab, this work was led by really fantastic graduate student, Ela Dye, and he was assisted by Jill Jaycox, an MD PhD student. But we've had just an incredible collaboration with my stalwart collaborator, Harriet Kluger, and her postdoc, Lilach Eisenbud. Thank you for your attention.
Thank you for that wonderful presentation. That is super exciting work, and I think very relevant to the work of this immunotherapy committee and also the translational studies and the samples that we collect. So I think there are potentially opportunities for collaboration. I think we have time for 1 or 2 questions. If there are any questions in the room for the audience, please go ahead and speak up.
If you have a question, please raise your hand. There's one there. Yeah, Kim. So is Aaron, are you online?
He is.
Indeed,
yeah.
Yes.
Thanks, Aaron. Margot, my question is there are 2 parts, which is how, I don't know if it's within a few minutes to explain how these findings are related to or are distinct from what I think Jed Walchuk showed a few years ago with the serum assays. And then part two of the question is whether these really reflect CD4 responses that are helping B cells make antibodies.
I'm really sorry. I could barely make out what you said. Could someone repeat or paraphrase the question who has access to a mic?
She will go to the microphone to repeat.
Oh, okay. Thank you.
Okay, can you hear me now?
I can hear you now. Yeah.
Oh, okay. All right. I don't know if it's a quick question or not, a two-part question. How and whether this work relates in any way to what Jed Walchuk, I think, was doing a few years ago with Cirex. And the second part of that is whether these reflect CD4 responses to tumor antigens that are helping B cell responses. Yeah,
killer questions, and great to see you, Kim. Jed did some great work on Cilex. I saw him at the PIC retreat about 6 months ago, and we talked about it. We see different sets of antigens than you see with Cilex, so it's sort of comparing apples to oranges, but they found some interesting responses that were associated with IRAE. We haven't seen too many IRAE-associated autoantibodies, with one exception. We found a novel autoantigen in about 10% of patients with hypophysitis. But in general, I think our data is indicating that for most IRAE, autoantibodies don't appear to play the major role, which I think is consistent with sort of the emerging consensus that it's more T cell driven. Your other question is, the other comment is very well made, which is that are these autoantibodies an epiphenomenon that are really reading out more on activity of CD4 cells? And that very well may be the case, but I don't think we can, I still think we can make use of that data. Meaning maybe an epiphenomenon, but without question, these antibodies are functional. Right? They neutralize interferon, they neutralize IL-6, they opsonize a tumor and promote ADCC. So the origins of these antibodies, I think, are interesting, but I don't think really impact our ability to make inferences about the impacts that they have on outcomes. But it's a point very well made. Thank you.
Thank you very much,
There's
Aaron.
another, Katie, there's another question here from.
Okay.
Go ahead, Nicole.
I think one last question. Yeah, go ahead, Nicole.
Thank you. Really phenomenal presentation. Thank you for bringing him here, Nicole Quidder, Seattle. In follow-up, really, Kim partly already asked the question about antibodies, autoantibodies and IRAEs. There has been great interest. Maybe the key question is also how well were the toxicity characterized? So many of our studies are still limited in really comprehensively and very detailed assessing patients. So I think we always want to be sure we have well-characterized data for that. So we'll be still very interested in potentially partnering with this large cohort that's getting larger, probably getting beyond 2,000 patients. We'd love to explore it together.
Yeah, that would be awesome. And I hope that we can get connected via email after this meeting. One thing I want to tell you about this cohort, Harriet's group actually collected it explicitly to look at IRAE. So actually, it turns out in this study, most of the patients, about 2/3 of them, had at least one grade 1 IRAE. So they're pretty exquisitely characterized for IRAE. That's why we did this analysis originally. And so we were a little disappointed not to see more associations with IRAE. And that's why, we said, well, what about response to therapy? And that's where we saw a lot bigger effect sizes. But I think if we go to a bigger cohort, we will be able to find more antibodies associated with IRAE. So yeah, I'd love to continue the discussion.
Well, thank you very much. So now we go back to the second session. We'll start with Katie to talk about the SWOG-IO agendas.
Yes, absolutely. Thank you very much, Siwen, and thank you very much, Aaron, for that wonderful presentation. So I think, in this session, we're really going to talk about some of the IO efforts across the NCTN network. And in terms of the SWOG Immunotherapy Committee, next. Someone can move. I think there are a couple of items that we've really been focused on. Yeah, you can show them all. That's fine. Thank you. I think that first of all, the SWOG Immunotherapy Committee really started in 2019, and it became as a research support committee. And in the spring of 2022, so a year ago, it became a full research committee. the main, I'd say the main sort of centerpiece effort that the Immunotherapeutics Committee has been working on has been working together with NCI on the development of the Immunomatch study. And in particular, most recently on the development of an initial pilot study that you'll hear about shortly from Dr. Huliskoven. This IMatch pilot study, actually, the protocol was activated in October, and it has currently has accrued 18 patients to the study. And as we're developing this IMatch pilot, now we're really gearing up for planning the broader IMatch trial in the future. One of the main goals of the Immunotherapeutics Committee is to facilitate IO trial design and inclusion and analysis of translational medicine in IO studies across diseases. And I think the example that Dr. Ring gave of the type of autoantibody analysis that he can do is something that is an example, for example, of a technology that could be incorporated into studies, into trials, and from which we can learn. And so this is one of our goals is really to bring information on these technologies and see how we can leverage them in SWOG in our trials. So how do we do this? We gather disease committee representatives with IO expertise, and we're working on that to really form an IO committee working group. So we've been reaching out recently, actually, to the disease committee chairs for them to nominate specific representatives to form this working group. And we already have some members, and we're trying to really fill that out and round that out and have that working group together. We hold monthly SWOG IO committee meetings to discuss trials and concepts. We just had a meeting last week, actually, which was a discussion on microbiome and studies of immune-related adverse events in clinical trials, and how we can study these and understand these in the context of some of the studies that are ongoing at SWOG. Next. So as I mentioned, one of the centerpieces of the work that we do is to host these monthly meetings on the first Friday of each month at 2:30 p.m. Central Time, in which, with our working group and with other members of the SWOG Immunotherapy Committee, everybody's invited. We discuss initiatives, people can provide updates on ongoing activities, and investigators can present trials and ideas for feedback. And we'll usually talk about one or two of these items at each meeting. If people are interested in joining and getting on the email list or presenting, please email either Chris Kippola, myself, and or Siwen Huliskoven, and they can be added to the distribution list for any questions. Next. So just to reiterate really the working model for the SWOG Immunotherapy Committee, this SWOG Immunotherapy Committee includes many representatives from SWOG. It has the co-chairs, representatives from the executive committee, from the statistical group. We also have this IO expert panel with members nominated from each disease committee. And I think one of the really important ideas here for the committee is that really we have broad expertise across different histologies. So our idea is really to cut across different indications and different histologies and think about translational medicine and incorporating that into studies and immunotherapy in the context of multiple diseases. And in fact, so how do we interface with the different disease committees? We interface with them in terms of cross-committee collaborations, developing IMatch and other IO committee agendas, and working on IO trial concepts and translational medicine design and execution. And then I think really importantly, particularly importantly for this conversation that we're having today, we also work together with IO committees and other groups, and we're going to be hearing about these examples and other efforts like the CMAC effort. And we also integrate, I think, with other groups that are partnering and that are part of SWOG, including the Jackson Labs, Cold Spring Harbor Labs, the SWOG Biorepository, and the SWOG Stats Center, and of course, SWOG Leadership and Operations. And we all work really closely together with them and with the NCI and the National Cancer Institute. So with that, I think I'll turn it over to the next presenter.
So next we have Dr. Cozono from Alliance. So he's the chair of IO Committee at Alliance. And also we have Dr. Mark Klein. Can you raise your hand? He's here to join us from Alliance as well, IO Committee. So David, do you have your slides? Do you want to share your slides?
Yes, happy to. So thank you for having me. Hopefully you can now see my slides. All right. So I in fact, chair the committee together with Andy Nixon, who is at Duke and really has a focus on translational medicine activities in our committee, a very much part of the heart of the work we do. We have an executive officer in Yuzhe Wen, who's also the director of the translational operations for the Alliance, so really well situated there. Ellie Deeb has been helping us from a community oncology perspective, especially as we found it very important to engage the broader network, including the NCORE sites when it comes to our studies and ideas. And then Laura Hoffman as our protocol coordinators really worked very well and hard with our committee. I don't have any real relevant disclosures for this talk, but I do have a consultancy on with Genentech. Roche, of course, produces IO products. So highlights about our committee actually dates back a while. We were initially established as a working group rather than a full committee, and this was back in 2017 when we were a working group within our translational research program. This was led by Beth Nithendorf at Dana-Farber, who led the transition from this being a working group to being approved as a full committee in the fall of 2018. We've been at this for a little while. And as of today, we have 74 members. It's a pretty large committee by Alliance standards, but as you nicely mentioned, Katerina, it really does take representation across all the different disease entities, modality groups, different specialists to really make a cross-modality committee like the IO committee work well. So that's what we've done. We've incorporated clinicians, researchers, but also nurses, statisticians, patient advocates, and administrators to really try to make our efforts as representative as possible. We have a portfolio of trials, both within the NCTN, but we also have, as I think many of you know, an Alliance Foundation Trials Group that helps with some studies in collaboration with industry for those that cannot proceed through the NCTN mechanism. We're very excited about our integration with your efforts on IMatch. Really look forward to contributing there. And then we also work closely with the CIMAX and work closely with folks like Dr. Wistuba, so I'm glad he's also talking today. I'll mention briefly about the iResist initiative, which we had some funding from the Alliance Foundation to try to understand one of the possible RESIST criteria that might augment what we can get from RESIST 1.1 in characterizing all the special characteristics of immunotherapy related response criteria. And then as you do, we also have monthly teleconferences where we speak on new concepts and ongoing efforts. I just wanted to highlight a couple of the efforts that are actually centered within our committee, because the vast majority of our IO trials are housed within the disease modality, specific committees rather than our committee. But one that's led by our group includes A15-1804. I would consider this to be a sister effort to your very successful iCheckIt effort, which I understand has now accrued over 1,000 subjects. We had as a somewhat different focus from the get-go, the intent of identifying patients specifically at the time that they experience one or more severe IREs, so largely grade 3 or higher, and focus enrollment of patients at that point, and then collect biospecimens and data inflammatory tissues when possible, but since biopsies are not always done, in fact, more often than not are not done, blood samples are a huge part of our biorepository, as it is with iCheckIt. We collect samples at the time point they come in with their IRAEs, and I feel that collecting samples at that floating time point is really important, because on most of our therapeutic trials, samples are collected with respect to different efficacy endpoints, whereas toxicity time points don't necessarily mesh well with those. We also collect samples 1 month after that initial presentation. The hope is that most patients respond well to corticosteroids, infliximab if needed, or other therapies, but not everyone does. And some people have progressive or refractory IRAEs that we want to understand, why that is. And so we have those 2 time points starting from the point of IRA presentation. But bringing this now closer to what you ended up having in SWOG, we recognize we need to have negative controls effectively, to be able to control, have control samples for people who've never developed these toxicities, fortunately. And so more recently, we did expand our protocol to include patients who are pre-registered, we call it, but basically can be enrolled even before they receive IO, so we could collect time point there. And we've decided to collect additional samples 1 month later, with the hope that we can see early changes in circulation in the blood, markers that might suggest the incipient development of an IRA, but to maybe identify them before they become clinically manifest. And so we now have these time points, 2 before and 2 after the IRA develops for comparison. It's very much my hope that this effort will be very complementary to iCheckIt, and that the 2 can really cross-validate the results of each other's findings. We do not have, like you, an NCTN-funded study within our committee specifically, but we do have one within the Alliance Foundation Trials group of studies. AFT50 is a study in endometrial cancer. This is where, like lung map, I see a few of you who've been on some of the SWOG lung map sessions as well, but this is now EndoMap, where we're similarly looking at Foundation 1 CDX testing to inform biomarkers to enroll patients on biomarker-driven sub-studies. And at this point, we have 6 activated sub-studies looking at markers that are commonly present in endometrial cancers. And as you can see on this slide, the vast majority are combinations of a drug that would be specific to a pathway such as like the PI3 kinase AKT-PTEN pathway, but then together with the PD-L1 inhibitor atezolizumab. The study isn't limited to just IO. This is in partnership also with the GYN subcommittee of experimental therapeutics in the Alliance. And we have a new cohort 6, for example, that doesn't contain an IO molecule within it. But we're really trying to find the best therapies for patients with these recurrent or persistent endometrial cancers. So it's an example of a study we're running, because really otherwise this cancer didn't have a home within the existing disease committees within the Alliance, and so partly how it came to us, that and its very strong IO focus. iResist is an example of an effort we're leading that isn't a clinical trial, but rather something in support of clinical trials. Recognize that Resist 1.1 really doesn't do an adequate job, I feel, of fully capturing in immunotherapy specific patterns of tumor response. Things like pseudoprogression, where you see radiographic increase in size of a tumor, but it really reflects the inflammatory infiltrating positive development on the way to having a therapeutic efficacy. And so there are a number of criteria that have been developed to try to encompass this a little bit better. iResist is one of them. The Alliance Statistics and Data Management Center folks have been involved in the development of iResist. So it's kind of, we chose to back this one in particular across our studies, had a small amount of funding to help develop protocol and case report forms. We could try to really broadly include iResist across our IO trials, to formulate tools to really help sites with it, because it's a little bit more complicated than Resist 1.1. And then resources to help educate Alliance and other NCTN researchers on how to use iResist. And today you can see that we do have iResist now included in about a half dozen of our active IO studies. Not every study that we have with IO, it doesn't make sense. For example, if it's an IO study of adjuvant therapy, because there's nothing to follow for a RECIST type criteria. And there are other reasons why by a tumor basis may not be the best. But we're really trying to actively encourage, and the IO committee reviews concepts as they're being submitted to the Alliance for suitability for something like this. And usually as a secondary or exploratory endpoint, because at this point, we recognize that this isn't fully vetted. But by studying it systematically like this, we hope maybe someday it could be. So I mentioned that most of our studies are, in fact, in IO are still run by our disease focused committees. And this just gives a flavor for what we have. When our committee started, all of the studies fit on one slide like this. Now we have enough to fit two. So here you can see that we have studies. These are some of ours that are in the lung cancer space. We have ones in rare diseases such as cutaneous cancers, angiosarcomas, mesothelioma, liposarcoma, and the like. And then we have other studies in GI, such as colorectal, gastroesophageal, rare genitourinary tumors, renal cell, urothelial, CNS lymphoma, and glioblastoma. So, many of our disease committees have at least one IO trial at this point, really highlighting the benefits of being able to coordinate with them on IO-specific considerations. These are just a couple slides highlighting a couple that we've had recently activated. If you're interested, all of this, of course, is posted at CTSU. I'm also happy to share details about some of our recently activated studies as well. And then just to mention again, the Alliance Foundation trials, we have to date had this handful of studies that incorporate at least one or more IO molecules, and the three on the bottom right now highlighted in yellow are those that are currently active. AFT 57 would be the newest to activate very shortly. So thank you for the chance to talk a little bit about what we've been up to within the Alliance. Here's some of our contact information. Really look forward to engage with SWOG and the other NCTN groups as we try to chart a path forward with IO therapeutics.
Well, thank you so much, David, for this introduction. We'll leave the questions to the end. So I want to ask whether Dr. Klein, Mark, do you have any comments in addition to David's talk?
I would say the only thing is we are starting a working group to coordinate with IMatch. I think we're actively thinking about how to bring up proposed new studies and whether or not they make more sense to try to eventually get into the IMatch platform or to try to do their own studies. For an example, I know that one of my areas of research has been in mesothelioma and for the cell cycle. And some of the folks have been trying to put cell cycle inhibitors and immunotherapy together, and we've had not very good success engaging companies on a one-off basis. So platform-based, biomarker-based studies across the board probably would make the most sense to get buy-in from the companies, because if the companies don't buy in, we're not going to get studies. So that's kind of where we are right now. So the other thing is I would say is discussing, the biomarker platforms to utilize. I don't know that there's a standardization around that yet. So.
Thank you so much, Mark. So we will have a panel discussion in the end. So we'll just gather all the thoughts together. Then I want to go to our next speaker from NRG, Dr. Ulla Hanan. She's going to talk about, she's the IO lead from NRG, and she will talk about their efforts.
So I'm very glad to be here, and glad to hear to everyone what experience you have. We are very new. So our committee really had our first meeting last week. We are very new. We've had a working group in NRG, but not a subcommittee. We are actually under medical oncology. So we've had our first real meeting, and very interested to hear how the other groups are working and getting ideas how we can further our work in NRG. So I had just, when Simon had asked me how we work in NRG. So we are an advisory group in the Immunotherapy Committee. We're not, we don't we can come with ideas, but it's the disease site committee chairs that do bring it to research strategy. And so the study chair proposes research ideas to the disease committee, and then the disease committee is what discusses and approves the idea. And then when that is approved, then the study chair can develop the concept, and it then goes to the research strategy committee. And once it's approved by the research strategy committee, then there is an sent to the executive committee for prioritization within the NRD research portfolio. So the research strategy committee has 4 meetings per year, and what we are trying with our immunotherapy committee is that we will have meetings 4 times per year prior to these meetings, so that we can look at the concepts that come in and have discussed and reviewed those concepts that have anything to do with immunotherapy, to be able to give feedback to the study chairs about how to incorporate the immunotherapy and if things can be done better. And so that's how we're planning to do things. And then in the research strategy committee, the chair and the vice chairs, so Janice Liu is my co-chair here in the immunotherapy committee. So the 2 of us will be are represented, and we'll review those the concepts and protocols that are presented. And we mainly look at what is the role of, because all of these concepts have been vetted by the disease sites. So what we mainly focus on is what's the role of immunotherapy? How do we incorporate it in this trial? Could it be done better? And mainly looking at also translational research and looking at can we find better ways of using the tissue and the blood that we are collecting. So it's very easy, the reviewers can add their comments. It's kind of a blank form, so we can put in whatever we want with the focus on immunotherapy. And then once a concept is approved, the protocol development begins with NCI approval, and then it circulates through all the study chairs, disease chairs, scientific core committees, and NRG staff. And so there are 4 circulations of each protocol. And we, as in the immunotherapy, we've asked to be involved early on, both in the concept, but also with each circulation of each protocol to try to give input about immunotherapy. So the role of the immunotherapy subcommittee, we are trying to have members in each of the disease core committees. And then so we can participate in the concept development. So we have about 20 members in our immunotherapy committee, and so we are trying to have members in each disease core committee and have those members actually review the concepts and protocols that they're most familiar with. And then, as I said, we participate in all protocol development circulations, mainly look at scientific content, eligibility, treatment, and key here, translational science. Yeah. And we currently have about 10, we have 10 protocols open with immunotherapy and 8 on concept level. So it's coming. So it was very needed in NRG to have a immunotherapy committee.
Thank you so much, Susanna. Looks like all of us have very similar vision and plans. So I will go to the last speaker of this session, Dr. Vysjuba. So he's from MD Anderson, and he's the lead of the NCI CMAC effort.
Thank you. So thank you for the invitation. I work very closely with in the SWOG group. I'm the PI of the biorepository located at MD Anderson. And because work that I do in molecular immune profiling. 8 years ago, I started an immuno-oncology biomarker group in the co-acting group, and became a subcommittee. And we have been actually working with investigators on the design of clinical trials, providing advice on biomarker, a lot of education on immune profiling strategies, and we had a teleconference in the old days, and then activities during the fall and spring eco-acting group meetings. And the entire eco-acting group is reorganizing how they approach the IO space, and even they have a early therapeutic committee that actually look at many of these efforts in immuno-oncology. They are going to have a special group like the one that you have, that actually will have probably the same goals and objectives, right? So that's why it's I recommended not to put me down as a representative of the eco-acting, because I do just some of the work, which is the biomarker piece. I suggested that maybe it's an opportunity just to refresh the memory to some of you about the CIMAC effort, which is an NCI-funded consortium as part of the Moonshot program that was established in 2018. These are my disclosures. And we have been working with different groups of the NCTN network on clinical trials. And I'll show you more about this network in a few slides. The idea of this network was an interest of the NCI to enhance mechanistic understanding of the action immunotherapy in combination in clinical trials, and accelerate the development of clinical relevant biomarker response resistant adverse events. Was initiated in 2017, and there were 4 CIMAC centers funded for this effort, and a central repository of data common center. And I'll explain those in a following slide. And now actually was renewed as a program. We reapplied all the centers, and we got the information that we will be funded. We haven't got the final letter of award, but we're planning what we call CIMAC version 2.0. And also this has a component that is called PACT, that is a partnership with industry, a partnership for accelerating cancer therapy, managed by the foundation of NIH, in which industry actually, they say that they put $10 million each, $100 million available for developing biomarker in the IO space. And they also, this company, they can bring their own clinical trial to this network. And it has been actually very good source of funding to put all this network together. So this is the org chart of the CIMAC CIDC. CIDC is the Data Common Center 1.0. Clinical trial comes from NCI or PACT through the CTAP, portfolio. And then we have the designated biobanks of the NCTN network, the group, part of the group bank committee. You have one in SWOG, I lead the one in EcoGAC, etc. And the samples come through them to CIMAC. And actually, the bank will have been funded to actually contribute to CIMAC. So it's part of our goals now and objectives. And then we have 4 CIMAC centers: Dana-Farber, Mount Sinai, Stanford, and MD Anderson. And the CIDC, the Data Commons, is in Dana-Farber, and they have created a wonderful structure with all clinical data and biomarkers and sample characteristics from all trials are getting there. And now we're actually trying to develop some strategy to do cross-trial analysis. And the version 2.0 is, the only difference will be that the CIDC will not be in Dana-Farber, will be in NCI. So some of the accomplishments, very briefly, we have analyzed sample from over 35 clinical trials on immunotherapy. We developed a tier assay system. So tier 1, whole exome sequencing, RNA sequencing, or nanochain IO signatures, cyto, multiplex immunofluorescence, and or immunohistochemistry from tissue, single place immunohistochemistry for PD-L1, and all in which study cytokines, chemokines, and growth factors in the blood, all those are tier 1 assay. They should be done in all samples from all patients, from all trials. Tier 2 is actually is selected for some trials and different approaches there on sequencing. Actually, TCR sequencing is going to move to the tier 1 assay. And then we have more tier 3, more innovative trials. There's a lot of work on single cell sequencing or spatial transcriptomics these days, but that's for selected studies. So we validate all the assays that we use in tier 1, tier 2, and tier 3. They need a validation plan. It's not a clear a test validation plan, but it's very strict. So they pass it up kind of approval. And that's painful, but it's good for our labs to do that. Also, we validate assays and we actually harmonize among different labs a series of assays. I have a slide about this, and we develop a specimen collection umbrella protocol that is actually available in our webpage. It's very detailed for whoever is planning to do collection for IO assay. These are the publication, the CIMAC-CIDC network published in 2021, 4 papers in which we harmonize immunohistochemistry, multiplex immunohistochemistry, immunofluorescence, a panel of CyTOF for PBMCs, and whole exome sequencing and RNA sequencing among different labs. So this is the summary of the 35 trials, over 2,000 patients, some from samples from different diseases, mostly solid tumor. We have 7 heme malignancies, and 80–90% of the trials include immune checkpoint inhibitors, no surprise. So I think it has been a challenge, but I think we established this network going to the next stage for 5 more years. And I hope, the standardization of assay, running a common set of assay in all samples from multiple trials, it will help us to actually develop some cross-trial analysis in the future and learn more about biomarker, not just by disease, but also in the context of therapy. That's my slide. And for SWOG, I'm very happy because I've been in the lung cancer committee for 15 years. We were able to finish the project with the S1400I cohort of the lung map, the squamous cell carcinoma trial, with a combination of anti-CTLA-4 and PD-1. And this actually paper is resubmitted in Clinical Cancer Research, will be published. And today, in the morning, one of our investigators presented data on the DART trial on rare tumor, the 1609, that actually is going very well. So happy to answer any question later. Thank you.
Ignacio and Mark, I don't know whether you want to come and join me. I want to give the audience some opportunity to, I hope our speakers online are still there. David and Susanna, and Katie is obviously there. So, to give our audience an opportunity to ask questions, both from a SWOG standpoint, but also across NCTN standpoint, whether there's any question about how to join efforts of all these immunotherapy focused studies or translational efforts. Any question, for example, how to utilize CIMAC. So any questions from the audience?
This is Katie.
We have
Do you have a question there?
we have a person coming up to the podium.
Oh,
Hi,
okay.
this is
Okay,
this
great.
is Dana from SWOG Ops, and basically I was puzzling over the different presentations, and at least in SWOG, there's a bit more siloing than I think there may be in some of the other groups. And, having the adverse event research being done through the NCORP grant and the treatment research being done through the NCTN grant. Now, obviously, all of the data are at the SDMC, so that can be shared relatively easily. But I wonder if we feel like we have the right people in the right places in the committees to be able to kind of cross-pollinate the ideas. And I know you've reached out to the disease committees, but I also wonder about like symptom control and quality of life, and having representation from the INCORP committees as well.
Yeah, absolutely, Dana. And we have been working with symptom control committee. So actually, we're helping with the translational part of that. Absolutely not just the disease groups, but also, for example, pharmacy committee. We didn't even know there was a pharmacy committee there, but it's actually quite relevant because a lot of the immunotherapy, the toxicity that associated with immunotherapy actually is quite relevant with the pharmacy committee. So we are actually reaching out to all the committees. And from the presentations, it seems like we're doing very similar things. Number one, try to be a liaison between our members and different disease committees and interface with other cooperative groups and interface with NCI. So it's very challenging because, as you know, it's not a traditional model like what Dana pointed out. It's how we initiate these efforts and how these efforts are going to be funded is still of question. And we need the collaboration from each disease group to do all these different studies. So I see Nicole coming to the podium, to the microphone. Yeah, please ask your question.
Again, kudos. Oh,
Before you
yes.
make your question, I would like to make a comment. I work very closely with ECO. I come to SWOG, and silos happen in all the groups. The smaller group probably it's easier for them. One thing that we suggest is help us, even if we don't have a committee, right? We have a subcommittee on biomarker, is that you need to connect with the group banking, with the biorepository, and invite people from the biorepository to be part of the discussion, because that is actually a point in the design and the trial that they can provide feedback. And they're not immunologists, but they can tell you, how sample can be processed and can be handled. So,
Right.
and then they will see everything coming through, because they need to approve the collection plan of the samples, right?
Yeah.
Great. And Nicole Kudera, Seattle, a comment and a question. Really, first of all, kudos to Dr. Liskoven and Dr. Politi. This is a really novel concept to, as you've hinted at now, bringing this by therapy type. And we are so excited to have you here and also help us really steer immune-related adverse events. And a big thank you also to David Cassano, who has been wonderful in collaborating already to trying to harmonize the two RRE studies, that we can validate each other's findings. I know there was first big hesitation if that's feasible, and David and other colleagues have helped make this happen as well. A big question for you about CMAC and about translational funding. How is the interaction? How does it happen? How do you get your funding if we bring the translational specimens? Do you want to kind of educate us how investigators should approach that?
Yeah, the trials come through us through CETA. Our role is to actually tell you about the CMAC, the network that we're available, but the trial need to come through them. So I encourage everyone to work with CETA and representative, and there are people from NCI that work closely with that team. Helen Chen is one of them, right? And put her in the spot. You can talk to her and get her excited about these trials, and then they come to us. What we do is we have a clinical trial working group that actually establish priorities on trials based on the therapy, the data on response, and then the availability of specimen for different assays. And then we have a committee that provide that priority, get back from NCI, and then come samples from the biobank to our laboratories, and the funding is provided by the grants. So it's free for the investigators of the trial, and then And then we work with the trial investigator. We have a lot of meeting on translational research based on different trials, and with the statistician from the group 2, very important, and for the analysis of the data. So I think that the system has been established and working relatively well. I think there is not enough money to do everything. So there's a selection process happening.
Gary, that would be the one last question.
Yeah, Gary Lyman with SWOG. Thanks for sparking this discussion. I think this is really good. And as Nicole indicated, we've had some cross-group collaboration with David and Alliance with regard to his study and interaction with what's going on here at SWOG. I just wanted to highlight what Dana mentioned, which is this need, despite our fledgling efforts, we're still very early in this whole development. We now have a full IO committee, which is terrific, and I think a bit ahead of some of the other groups. The IRAE effort actually sprung out of a request from Chuck Blanke to me 5 years ago to pull a group together and start these discussion. And Krishna and Nicole and others came together. Now Siwan's involved with the I Check It study, and that's gone very well. But I just want to emphasize this was kind of grassroots effort, and it's not a big surprise that the cancer control side sprung up with interest in the toxicities, and NCTN side, the IO, in terms of therapeutics. But we do need to find a way, even though, again, I think we're a little ahead of the ball, to bridge and bring these groups together, because we feed each other. These trials obviously produce both efficacy and toxicity, and we need to better identify the patients at risk or at benefit across these. So anything we can do to better bridge this, and I think this is true for all the groups, this is the challenge ahead now, how we work together across the spectrum of outcomes for patients being treated with these therapies, which is obviously the future of much of oncology right now. Thank you.
Yeah, thanks, Gary. I think we all realize we need to work together, and I think this meeting, this session is a good start. You know, Mark, David, Ignacio, and Susanna, I think we should have representation in each other's committee and just increase collaboration and work together. So thank you so much for this great session.
Thanks for having us.
So, Katie, do you have
Thank
any closing remarks for this session?
So, I mean, I think the presentations and all of the comments, I think, have been really helpful and valuable. I think, you know, any efforts that we can have to increase collaboration and communication within the groups and then between the different groups working together is, I think, going to be really helpful in getting to know each other more and what we're doing. So, you know, maybe we can think of having even quarterly meetings between the different IO committees from the different groups and just sharing what we're doing and talking more. I think those are things that we can discuss.
Thank you, Katie. So our next speaker is our lead statistician at SWOG, Dr. Michael LeBlanc. Everybody knows him. And there are a lot of questions asking, you know, how can we utilize a SWOG database? And he's going to talk about it and tell us today, especially related to IO studies.
So I also realize we're a little bit, I think we're a little bit behind time,
Yeah.
time here.
That's okay,
So
Michael.
let's see if I can get this in the presentation mode. Okay. Oh, okay. Thanks a lot. Yeah, so you know, following all the science and new trials development, so I'll veer to a little bit of the boring side here. But I really just wanted to follow up the other comments in terms of collaboration. I think what the couple of simple tools and opportunities I'm going to talk about really, I think, are just a first step in facilitating that collaboration. So what you can find just looking on the SWOG website, if you're interested in studies that have already been conducted and you're thinking about analyses or projects that could be related to data that we've already collected, you know, the IO portfolio is growing based on individual disease committee work. And so just take a look at what's available. And that's going to be my goal, and I'm going to try to do it a little bit faster than I planned, because we want to talk about IMATCH, because there's lots of good stuff to talk about IMATCH.
But this is important, Michael.
Okay, so data type is I'm just trying to get myself off the stage here quickly. So data type, so we're interested in obviously in national trials that may have used immunotherapy. And it's always about addressing a question, right? They always think that you're kind of saying the obvious, but sometimes it gets lost. The focus on the question, I'll hit back. Oh, and a sidebar here, I'm going to advertise the TM plenary session, which I know everybody's going to, because there's a statistician talking at the end of that, Megan Othis, who's going to phrase she thinks some of the best ways of interacting with statisticians with respect to translational medicine studies, and I think they apply here as well. So I'm going to talk briefly about how you might find whether there's data on SWOG studies that have been conducted that may have some data that might be of interest. SWOG's not the only group, as we've heard. There's a lot of the NCTN groups. And there's data sharing associated with them. And I'm sure you're aware of the NCTN NCORE data sharing archive. And now as these studies have progressed, those studies are phase 3 studies, and some of them are being uploaded. And we spend a lot of work at all the groups to make sure our data are good to upload to the NCTN archive. So we should utilize each other's data. So, and then finally, I'll talk about the Hope Foundation support for secondary analyses. We're a committee, but I'll tell you, we're always stretched in terms of statistical resources. So if you think of a good proposal, especially that involves across studies, a great way to fund that is through the Hope Foundation, who supports us to support statisticians to work on cross-study analyses. So how to access the data, and the slides are available afterwards, so you don't need to pay too much attention. So directly, SWOG has a data sharing policy, as the NIH has a data sharing policy. We all do. So you can access the data for your own use or work with SWOG statisticians. And so can everyone else. But the NCTN data sharing archive is a very specific archive that we've been building up. The groups have been building up since 2015. And I know several phase 3 randomized studies involving immunotherapy are now uploaded in that across the groups. And then those data are curated in the sense that the data dictionary is available, and the process for accessing them. And I've used the data sharing archive myself from another group's data. And so I know the groups would love to have as many people use this as possible. But on our own website, we had a session, actually probably just before COVID, and people said, well, what about the SWOG data? How can I find out what's going on? And everybody's probably seen the clinical trial search tool, but what we did was we went back and said, oh, let's add a couple extra search features to see whether, to help select data from that. And so here I just show the web page associated with that. It indicates on the first website how many patients' data actually have been enrolled in SWOG studies, a couple 100,000 over the years, how many open trials, a lot of them now involve immunotherapy, one sort or another. And basically the search tool, this allows you to search on simple features associated with that trial. And so here's the search engine. And you can fill in things, for instance, the phase of the study, whether it's open or closed. You're likely not going to get access unless you have approval, but you won't get access unless you have Data and Safety Monitoring Committee approval for an ongoing study. So likely closed studies. On the side there, you'll see registration, whether they're pro forms. That can be important if you're looking at outcomes related, pro-type outcomes associated with immune-related toxicities. There might be a secondary question you want to go and think about banking data. We have other data available banking, and that's I'm going to do a sidebar here before I forget. Each one of those disease committee chairs are presented with what's in the bank associated with every study before the group meeting. So in terms of a collaboration of the study, if you find a study that's associated with them, go to that disease committee chair or members in that disease committee, and they can help you plan together and think about what studies might be relevant there. Some of those samples are probably spoken for by existing things, but some of them might not be. And so in this case, I just, the next one, I just typed in nivolumab. So I knew something would come up there. Actually, the study that came up first was, of course, 2101, which is what we're going to talk about next. And it has, it's obviously has modest approval. It's probably not something you can request the samples are out of right now, but it just gives you a sense of what the search is. Simple searches. You can search for other terms in there as well. And then in this case, as I said, it was search for the 9 trials came up. You can refine the search, and here I refined it to phase 3 studies that are closed. And so with nivolumab, and it came up with lung map study S1400I, which is actually on the NCTN data sharing archive as well, includes PROS, has all sorts of good things associated with it. 1826, which is just closed, which is a study in the lymphoma committee, which you'll hear more about shortly. So this is examples of the search. But the other part of it, I also want to say, SWOG is not alone here. And so this is, and I wanted to highlight the data sharing archive. So these are limited to phase 3 trials. So unlike the SWOG data sets, it's a little bit more limited in terms of those large phase 3 trials. But there's definitely immunotherapy trials in the archive now because of the timing of this. And so they're in that data structure. One can then make applications for the specimens associated with that. It's just a little bit more work. And there's some examples of studies that were in the database a while ago. But I also just wanted to end off with access support for doing those secondary analyses. You're not restricted. You can make a request, do it outside of SWOG. That's fine. But if you want to do it within SWOG, one of the mechanisms the Hope Foundation has generously funded is statistical support for cross-data analyses, and it's an opportunity I think that's underutilized by the groups. It comes with an application. It comes with formulating a question and obviously a well-paid. But the submission is a lot less than a grant. So you can make requests there to do statistical support from us to do some of these cross analyses. And as I said, it's a short proposal, and I think it's only appropriate. The Hope Foundation's 30th anniversary. They do so much for us as a group, and this is just one example of where they support us. The final comments, the Hope Data Analysis Program. Remember, this is what it is. One needs a question that's articulated in a way that you're going to buy a few reviewers in there to make sure that that's a well-formulated question that's feasible. We can help you with that in the Immunotherapy Committee. SWOG data available, of course, not from ongoing trials typically. The NCTN data archive, as I said, is another source of data. And if nothing else, it tells you who to collaborate with to make this happen. It's not necessarily you need to use this resource entirely, but it tells you who in the groups have these data that you need to talk to. I just my line here. It takes a heck of a lot to collect this data, so we're really enthusiastic about it being used in other sources and understand what's going on. So high speed, but I just wanted to give you a flavor what was available and hopefully to start the conversation with other groups.
Yeah, thank you so much, Michael. You cleared a lot of questions or mystery that we had before, and it looks like a very straightforward.
Right, it's fun.
Yeah.
Actually, sorry, I would cut you off. It's that seems like a really simple add-on to some of those search engines in the SWOG website, but it is kind of fun to see what the portfolio looks like,
right,
even outside of immune therapy over time, where the data available.
right. So before Michael escaped from the stage, does anybody have any question for him?
Everybody wants to know about iMatch.
It's very clear. Okay, thank you so much. So we'll go to the next session. We have 35 minutes left to cover this. So many of you know the iMatch concept quite a while because we have been presenting this for a long time. And I'm I'm glad I can provide an update today with actual data from the trial. So, just a very brief summary of the IMATCH effort over the past several years. It's a huge effort involving multiple branches from NCI and almost a lot of the important groups within SWOG and a lot of people from SWOG operations, the stat center, the central bank, and took several years. So we started in 2019, just put the concept together, and the concept was approved a year later, but it was recommended to run a pilot because the structure was kind of new, and we're not sure whether it can be executed or not because it involves prospective biomarker identification or so the IMATCH concept was brought up, and it took 2 years to go from a concept to approval of the protocol. So finally, October 14, 2022, we opened the IMATCH pilot trial. So what is the IMATCH concept? Just escaped. So we all know, it's the immune resistant mechanisms is heterogeneous, and a lot of clinical trials are failed, not because the treatment is not working, but because the heterogeneous nature of our patients towards the resistance to immunotherapy. And we all know we need a biomarker to do precision oncology or oncoimmunology to find the right patient, find the right treatment for a particular patient, but we're not there yet. So the IMATCH concept is to utilize available biomarkers, not to try to predict a response or resistance, but use the biomarkers to separate the patient into predefined molecular groups of similar kind of resistant mechanisms. So, to help combination immunotherapy to find signal and identify subgroup of patients to treat and move forward with. So this is the IMATCH structure that was proposed. So there is a central screening protocol, and my co-chair, Kitty Politi, is the chair of this central screening protocol. Every patient going into, regardless of the sub protocol, every patient going into the platform is going to be profiled. Their tumor will be profiled with whole exome sequencing and nanostrain IO360 to separate, group them into these biological groups based on these biomarkers. And individual therapeutic protocols will be developed under that. So to have such a huge undertaking, we need to know whether this prospective biomarker profiling is feasible or not. So we have this pilot study to explore the feasibility, whether we can do that or not, and also streamline the central protocol operational structure. So here comes the S2101, the Picasso trial. So we take patients who have advanced melanoma and head and neck squamous cell carcinoma, and who have already progressed on anti-PD-1 and treat them with cabozantinib and nivolumab. And all patients will be profiled and will have equal number of patients in each biological group. And the goal of that is to look at the feasibility of this approach. So basically, patient who fit the trial profile will be consented, and they either have archival tissue or they have fresh biopsy, and the site will submit the tissue before enroll the patient into or after they register the patient into stage 1. So in stage 1 of the trial, all patients will proceed, regardless which group they belong to, will proceed to treatment. And there will be a temporary hold feasibility analysis after 45 to 60 patients are enrolled. And if it passed the feasibility criteria, then we'll enter stage 2, where patients actually after the biomarker profiling, they have to wait for the result, know which group they belong to before they can actually be treated. There is a possibility if the subgroup is full, all patients are planned patients are enrolled, then they could be a screen fail. So in the end, we'll have 120 patients treated, 60 melanoma, 60 head and neck squamous cell carcinoma, equally distributed to these biological groups. So this is just a summary of the study procedures. And these are the objectives of the pilot trial. And again, the main goal is to look at feasibility. And the most important part we need to focus on is the primary endpoint is the tumor or the biomarker turnaround time. So it counted when the patient sample is submitted by the site, and it ends when the site gets the notification of the biomarker assignment or a biological group assignment. And we are looking for at least 75% of the cases will be reported within 21 days. And we're also looking at the clinical outcome, the response rate in each of these biological groups, and we're looking at the safety profile as well. So the biomarker, it's the subgroup assignment is based on cutoff of TMB, tumor mutational burden, and the nanostring TI score was a preset cutoff of TMB of 10 and the TI score of 6. We will look at the feasibility of this at interim analysis. So these are pretty standard eligibility criteria. I'm not going to get into the detail of that. But basically, we take patients with advanced melanoma, head and neck squamous cell carcinoma, who have already experienced anti-PD-1, and that they have either archival tissue or fresh biopsy to be submitted. Then they will be eligible for this study. And there are some study-specific criteria. For example, there is some stipulation about controlling the blood pressure and the prior amount of radiation therapy, and whether there is a risk of tumor bleeding for head and neck cancers. And the patient has to be able to take oral drugs. And the one important thing is the pills cannot be crushed. So this is a study calendar. I will not get into the details, and there are a lot of built-in feasibility analysis, interim analysis to ensure the trial is operated in the optimal way. So here is the real data. As of April 30 last month, the end of last month, we have 114 sites that are activated. We have 18 patients that were registered to step 1, and 17 patients actually get treated under the protocol. There's one patient was enrolled from ECOG and one patient from NRG. And the map on the right side, which is put together by my friend Katie Campbell from UCLA, shows the different sites that enroll patients and where they are. And the stars tells us where all these central labs are. So the red star is where the central bank is. The blue star is where MOCA is, where they will process the tissue of the nucleic acids for TMB reporting. And the orange star is where our stat center is. They get the information, the report, and send the report to the sites. So how do we calculate turnaround time? It starts when the sample is submitted by the sites. It usually takes 1 day for the tissue to arrive at the central bank nationwide. And nationwide will extract process the tissue, extract nucleic acids, including both DNA and RNA. And then if it meets the criteria, then they will ship the DNA to MoCA lab, and MoCA lab will process the nucleic acid, process the DNA for TMB calculation. And after that is determined, they will send the notification to SWOG Stat Center, and Stat Center will send the result back to the sites, and that complete one turnaround time cycle. Right now, we don't have the approved TIS assay ready yet. So we're not counting that time, but it is anticipated it's going to be much less than the TMB reporting. So far in the trial, we have data from the first 16 patients, turnaround time. And out of those 16 patients, 9, the number actually is wrong. Oh, yeah, it is. 8 is melanoma, and another 8 is head and neck squamous cell carcinoma patients. And the blue dots on the left are the total turnaround time. So the first set of data is the days the tissue stay at nationwide. Second set of data points are days at MoCA lab, and the third set of data points is turnaround time. And that there are a few samples that failed the initial process and was reported not enough nucleic acids. So on the right is a figure showing us more detail of each sample. So it's actually sequenced from left to right, the sequence when they are processed. So the very first sample in the trial is number 1, and most recent one is number 11. The red bars are melanoma samples, and the blue green bars are head and neck squamous cell carcinoma samples. And the ones that labeled with star are fresh biopsy tissue. So you can see it's actually a mixture of melanoma and head and neck squamous cell carcinoma, and we actually have quite a bit of success with fresh biopsy. But there are a few samples that fail the process, and we'll talk about those. And you can see on the right, most of the samples meet the criteria. The turnaround time is either at or within 21 days. And then the central lab and MoCA are working on reducing that their respective timeline to make that number even smaller. So what is usually the reason a sample fail? Usually is insufficient tumor content, the common reason is a IR guided procedure, and also it seems like head and neck seem to have more failure than melanoma, and also end of week shipment usually associated with longer turnaround time. So there are some actions that are already taken to try to maximize or optimize this process. So the central bank is going to embed all 4 fresh cores collected in one block to increase the nucleic acid yield. MoCA lab lowered the minimum requirement of nucleic acid for TMB testing, so that more samples can be processed and can be successful. So also we are trying to engage site staff and their interventional radiology teams, so make them aware this is a very important process. And also we're preparing a biopsy manual, including pre-biopsy scoring schema to predict the likelihood of success, trying to maximize this, optimize this process. So the frequency of specimen failure is already decreasing. So my colleague, my co-chair, Dr. Paul Sawicki, put this slide together just to try to emphasize how important it is, to follow the protocol for biopsy, for fresh biopsy. So the just from our analysis of the existing procedures, existing samples, it looks like if a tumor has a lot of necrosis, then it's very difficult to get sufficient amount of nucleic acid. And the central lab prefer Monday to Wednesday shipments. So these are more operational, but it actually helps to reduce that turnaround time because the weekend can be in the way. There are some steps that central lab actually recommend to avoid internal delay within their central bank because they have a process. They provide a shipment label for this study, and because the biopsy and shipment actually is paid by the study, so they have a special process. So make sure to use the shipment label provided to ship the samples to the central lab. And most importantly, engage the IR radiologist before the procedure is done and communicate the requirement. The protocol actually require 4 cores for the fresh biopsy procedure. Each of them should be at least 1 to 2 centimeter long, and the needle should be 16 to 18 gauge. Also central bank recommend consider placing those 4 core biopsies in BioWrap as a kit that they can provide, and it can help to reduce the fragmentation of these tissue. So there is a table on the right that actually is used in NCI match. It helps to estimate what's the likelihood of success of certain procedure. And there's a certain criteria. There's a list of criteria you can use to estimate the likelihood of yield. And if it is less than 25%, the advice is do not proceed with the biopsy. The likelihood we're going to get enough nucleic acid is actually quite low. And of course, if it is more than 70%, proceed with the biopsy. And if it is 25 to 75%, make sure to engage the IR radiologist and the team to make sure that sample is collected in the most optimal way. So there are some frequently asked questions. Do we have to wait until molecular results are back to register patient to stage 2? And the answer is no no for stage 1, but yes for stage 2. So remember in stage 1, it's 45 to 60 patients. So that's where we are right now. And the patient don't need to wait for the results until proceed to treatment. But in stage 2, after the interim analysis, we do need to have the patient wait. And also, can cabozantinib be crushed? And the answer is no. Who should I contact with protocol question? And this S2101 question email is the most preferred because the entire study team get the question. So again, it's a lot of people's efforts to put this together. I'm so glad for the first time we're able to provide data from the trial and some success, something we need to work on, and we'll continue to work on it. So I'm going to turn the stage to my colleague, Maggie Goodman. She's the study coordinator at Huntsman for this study. So we have enrolled 6 patients to this study, including both melanoma and head and neck squamous cell carcinoma patients. So she's going to talk about some of her experience, how to survive this protocol, what's the some success, some experiences, ways, tricks that she can use. She has put together to make sure the protocol is followed.
Thank you so much for such a warm introduction. So I'm Maggie, and I'm a CRC on the melanoma thoracic head and neck team. I started at HCI in June of 2022, excuse me. As Dr. Huliskoven said, for the SWOG 2101 trial, we were the first to open and the first to accrue a patient. So far, our site has accrued 6 patients, 4 in melanoma and 2 in head and neck. I know this trial is complicated, and I'm really happy to share our experiences with the group. Just to note, I don't have any COIs to disclose. To get these biomarkers assigned, there are some pretty strict tissue requirements. Tissue and cfDNA tubes are required to be sent within 24 hours of step 1 registration. And the exception would be for fresh tissue biopsies. Those must be sent out the same day of collection. This creates a really big timing and logistical issue, such as if tissue is being pulled from another site, you would need to allow for enough time for the request to be processed and the tissue to arrive. It's very important to have providers that understand the timing and the constraints of the trial to help advocate for your patient. So this is our study flow if archival tissue is available. The investigator would identify a potential patient. The CRC would run an archival tissue check to confirm tissue meets requirements. To note, a tissue check is possible at our site prior to consent due to a waiver that allows this as part of the pre-screening process. Tissue checks would only occur if the tissue is a mandatory component of the trial, and we are only notified of the tissue quantity and availability. No other information is available. If the patient is interested, the patient would then consent. We would do any of the step 1 screening to determine the patient is eligible. And then an adequacy check would be performed by our pathologists on site. Once the tissue is in hand, which would mean processing it into slides or the cores are prepared and ready to be shipped, I would prepare a screening appointment to ensure those labs are performed, and the patient would be registered when they arrive in clinic, and tissue would then be sent out to meet that deadline. Some keys to success here. Our team meets with specimen processing, our program manager, and the provider to discuss the workflow for each patient. An adequacy check is performed to ensure the tissue is sufficient with enough viable cells, and this ensures the tissue we are sending is high enough quality to meet the endpoints of the trial. This workflow has become key to ensuring we are doing things in a consistent manner and the process is as smooth as possible. So some notes on adequacy checks. I met with our pathologist at the Huntsman. Trial requirements, the surface area has to be 25 millimeters squared with a minimum of 5 millimeters. This requirement is tumor content and not referencing the total tissue surface area. And it is key that a process and workflow is in place to ensure the pathologist reviews this tissue. Something she also pointed out is that biomarker testing for eligibility is becoming more common for trials, and it's key that the pathologists are involved from the start to ensure smooth workflows. All right, so this is again some more information on the adequacy checks. So I would put in a request for archival tissue check. Our specimen processing team would place an order in Epic to request the pathologist reviews the block. Research histology would send the review form and the slides to the pathologist. The pathologist would then review the tissue to determine if it meets criteria, and then specimen processing would contact us and let us know if the block meets those requirements. Once the patient, once the patient and block is ready, the CRC will register and the tissue will ship same day. So we go through that same process. So fresh tissue biopsy, this would only occur after a tissue check confirms that tissue is not adequate or it does not meet the collection deadline of within the past 6 months. A patient would consent. We would then coordinate with the biopsy coordinators to schedule the biopsy. Patient would be deemed eligible. They would arrive in clinic for the biopsy, and we would register them to step 1 same day. Then we would confirm that the tissue was collected and prepared to ship. And then we would ask that the patient returns to clinic for the rest of the study labs and screening. Some notes here. We still perform that same meeting for these patients. And this is to confirm the site of the biopsy and make sure all of our workflow is worked out. Biopsy lead time at our site can be up to 4 weeks. Image biopsies also require recent scans and specific labs to be performed prior to biopsy. Due to trial constraints, this could potentially throw out existing scans or blood pressure measurements and other trial eligibility requirements. And again, it's important that the provider can help in moving things forward for this patient. So after step 1 registration and after the tissue has been sent, like I stated, we would perform the screening visit. The patient would be deemed eligible for step 2. We would register them to step 2, and then the patient would have to start treatment within 7 days. Okay, some pitfalls. Here at the HCI, we have a very large geographical area that our patients travel from. For patients that travel from out of state with an early biopsy time, I would confirm that the patient has arrived in Salt Lake City and register perhaps the night before the biopsy. Patients also have lives, and that makes scheduling difficult, which might increase lead times and highlights the importance of really good communication. For the fresh tissue biopsies, the provider also must complete the operative or radiology notes, and that has to be sent with the tissue. So it's important that when you schedule these biopsies, you communicate with the provider that the note needs to be completed prior to the shipping cutoff time. If pathology adequacy checks are not completed, again, we risk the tissue not being adequate and testing not being performed or being further delayed. There's also the added complexity of working with multiple teams and medically complex patients. And after each tissue is enrolled, we meet again with the specimen processing team, the provider, and the program manager to review what went well and what we can improve on for the next patient. And that's all I have.
Thank you so much, Maggie.
Thank you.
That was very helpful. Let's see. Yeah. So I know we have quite a bit of study, activated site study teams and study coordinators online. And we also have Thomas Butler, Susie Butler from University of Michigan, and Gail Kent from St. Charles Health System, in Oregon, joining us online as well. So if anybody has any question about the protocol, please go ahead and ask, because these are study coordinators from high enrollment sites, and they have a lot of experience with the protocol.
Steven, hi.
Yeah.
Thanks so much. First of all, great effort to lead a biomarker IO-specific trial. My question is more about the logistics. As all of us know that these days, staffing shortages are very common at cancer centers. So when you have a trial like this that involves multiple complexities, multiple teams, is there any thoughts to decentralizing the tissue assessment for TMB and gene expression profiling? So what I'm trying to ask is, MoCA does a great job, no doubt, but there is other companies, Foundation, Tempus Caris, to name a few, that have RNA-based testing incorporated into their platforms. To make something like this easy and a little more less complex, could that be an approach? Because I'm trying to understand, it's great to see feasibility and seems like it's coming together. But when you add the complexity of so many teams, especially with the staffing shortages that we have seen post-COVID, it does increase a lot of burden on the people that are trying to manage all the additional stuff that they have to deal with. So are there any thoughts on that standpoint to, not just partner with the NCI, because this requires a very strict effort, timeline-wise, team-wise, versus trying to decentralize it and bring it up for discussion with some of these other companies that could make this easier, way simpler?
Yeah, Rafi, that's a good thought. And there are several reasons. First of all, that's NCI's preference, right? It's that's not something that we can decide. But I think there are several reasons that we're not doing that. Number one is the TMB reporting is kind of heterogeneous across different platforms. You're referring, what if the patient already have TMB measured? Why do we have to do that again, right? And that depends on when that tissue was taken. It could be before the patient ever been treated by IO, when they were IO naive. That's actually not eligible for this trial. And we prefer, we actually want to check the tissue after they already developed the resistance, and within 6 months of that, right? So it has to be a very specific time point, and it's become very difficult to find, similar kind of tissue, and also acceptable TMB reporting platform, right? So it become a very heterogeneous reporting and the results system. So
I guess what I was trying for, let's ask, was, if you apply the same rules potentially in the near future with the same, cutoff for when you get the biopsy, but you use or outsource it to not just one entity, but multiple entities.
Yeah.
And I know like the NCMO specifically has participated in TMB harmonization with some of these companies through Friends of Cancer Research. So could that not be a futuristic approach to make this more decentralized?
Yeah.
Simpler and easier?
I think that's definitely something to consider because of the complexity of getting all these results, right? But right now, because it's a pilot, because we're trying to test the structure, the process, we prefer to do everything in a controlled way. Yeah.
And I think this is Katie, just to add something. I mean, I think one of the things for this pilot is really to test the feasibility of this approach also, which, would then be incorporated into the broader IMatch study. So, I mean, and one of the questions is this doable along these lines? And hopefully we'll be able to answer that with the pilot.
Thanks, Katie.
What is the minimum requirement for TMB testing? Are you approaching that with, nanograms of DNA available?
Correct. Yeah. So that's one of the approach previously. NCI was asking, MOCA was asking a lot of nucleic acids. Right now, they lowered it. I think it's 160 nanogram.
160?
160 nanogram.
That's a lot.
Acid.
That's a lot.
Yeah.
I mean, you can do a whole genome with 50 easy.
Right. Yeah.
Okay.
There's a question online. There's someone who has a hand up. Jasmine Davis. Hello? Jasmine, do you have a question? Maybe not. Maybe we can go back to Jasmine if, later.
So I want to invite is Gail on the online? Gail Kent?
I am. Can you hear me?
Yeah, Gail. So Gail is from a Uncorp side, and she has done a fantastic job enrolling patients and make sure the tissue are, up to the standard. So Gail, do you have any experience just from the community side angle, to deal with the complexity of the protocol?
Yeah, actually our workflow is similar to the one outlined before, and we pair with like a third party pathology company. So we can also, through our pre-screening process, check the quantity of material in a block that's on site already, which is great. I'm actually just going through the process of screening our first patient that'll need a fresh biopsy. So that's good to hear the tips actually and the insights for that. I am dealing right now with making sure that we are communicating and coordinating with the IR team. And that pathology and operative report is my one hangnail right now. So just, getting on top of things early, I think is important. I would just say too that just a couple of things in preparation for this, thinking back, just to know that the palliative radiation isn't allowed on trial. So just to make sure that you're communicating with your provider ahead of time. If they want to do anything like that, it needs to be before enrollment, and ensuring that washout period is looked at, taken into consideration. And we've had some hypertension issues as well, and getting that through this after consent screening process. So it's just something to really look at with the cabozantinib. That's been kind of my experience. Tips.
Yeah, thank you so much. Thomas from University of Michigan, do you have any comment? So University of Michigan is one of the highest enrollers.
Sure. I would just second and third that you need to have a very good workflow. I'm like Susie Butler, no relation, who's I'm on the data side. Susie's in clinic. She's busy right now in the clinic. That's why she can't be here. But like, I really rely on her just to have, like, we consented one the other day for, it'll be our first melanoma. And like, it's well in advance. You have to have everything kind of planned out in advance, because that registering and shipping everything same day and getting a biopsy and shipping all your specimens in the same day, it can be a real bear. So it takes a lot of coordination. And we're actually we've had a few issues with our first few patients where we weren't getting enough DNA in our specimens. So that Dr. Swisicki's slide, actually, I just saw there. That's good info, and that's what we've been working with to better at our site, is to work with IR and communicate to them like how important this is. Not that they're not doing a good job, obviously, but like how important it is that we get a viable specimen. So it just process, and just really having everything, all your ducks in a row before you move forward, because it can be tough.
Yeah, thank you so much, Thomas. It's I know Maggie has a very good workflow, and it's amazing. She knows all the detail and what's the time point that she can do certain things, and it's the same for each patient. Once you have that workflow, I think it's going to be helpful. And since we have high enrollers, I would volunteer, Maggie and all these CRC that participate today. If any other study sites have questions, just reach out to us, and we're here to help. I think it's just the initial stage to get familiarized with the protocol, and then everything will go smoother afterwards. And again, the tissue collection is a very important part of this study. So for archival tissue, a pathology review for adequacy is quite important before the tissue is cut. And for fresh biopsy, the size of the needle, the length of the core, and how many need to be followed per protocol. And communication with the radiation oncologist, interventional radiologist is very important if it is a IR guided biopsy. So, Katie, do you have anything you want to discuss?
No, nothing in particular. I think thank you very much to everybody who spoke today and for everybody's perspective. I think it was all very interesting and very helpful. And I think especially with the trial, to reach out and hopefully we can answer any questions that people have regarding the study and the specimens. And I would add that things evolve. So as we learn more and we learn about the hurdles, we are making changes and trying to address them to make things easier. So please let us know so that we can address these issues.
Yeah, thanks, Katie. So this is not just troubleshooting for this trial, there's a troubleshooting for the iMatch platform. So I appreciate everybody's participation, help, and patience. So I think that concludes this session, the Immunotherapy Committee meeting. Thank you so much for joining us. And we have a monthly meeting. So, you know, if you are if you're a member of our committee and you're not receiving the meeting invites, please let us know. It's a good platform to talk about initiatives and discuss agenda in our committee. All right, have a nice rest of the meeting and safe travels.
Thank you, everybody, for joining.